capture the regional variability and reduce whatever influence there is of the local factors, we need a lot of specimens to be able to do this. We are trying, in both the case studies I'm showing you today, the replication approach. So the collection we used, we had 29 samples from several islands across the Aleutians, going from Attu Island in the far west over to Akun Island in the far east. The samples were collected live. Some of them were museum specimens that are archived at the Smithsonian Institution, and then some of them were collected during cruises in 2004 and 2008. We dated them using growth band counting, and then for the longer specimens, we independently verified the dates using uranium thorium, which was one of the ways we were confident in knowing that the growth bands were annual. And so these ones have been uranium thorium dated. And then what we did is we scanned them, we polished them, we imaged them using a very high resolution drill that was attached to an XYZ stage, so we could take about 100 images and then stitch them all together to get one very, very high resolution image. And then for a lot of fun time, I identified all the growth bands in the skeleton, and then we measured the widths of the growth bands. And so we measured the growth rates, and we would measure them in multiple places to try and really constrain. Obviously, there's some variability in the width of the growth band across the algae, so we would try and do multiple transects to reduce our uncertainty with the counting. And we did this for all of those 29 specimens. And this is one of kind of a bigger cross-section image, and this is material removed for the uranium thorium dating in one of the live collected specimens. So when we take all these data, we kind of end up with a lot of data points on our graph. So we have year going from left to right with anomalies of algal growth. Sorry, they're normalized algal growth on the y-axis. And then with the individual samples are in light gray, we averaged, and then we filtered it to try and extract out that longer-term decadal, multi-decadal trend. And I'm going to first focus on what's driving, how we determine that the Aleutian low was kind of the primary influence on the growth rates by looking at just the past 100 years. So here, going back to 1920, we have the 11-year mean of the average growth. And I'm going to compare that to, first, sea surface temperature. And you can see that there is no relationship between growth and sea surface temperature. This is not true in other locations where there is a stronger seasonal cycle. But for this site in the Bering Sea, we see that there's no relationship with the seawater temperature. We compared it to the Aleutian low. It's the inverse, where the weak Aleutian low is up and the strong Aleutian low is down. And we can see that it significantly correlates. It's not a perfect reconstruction or a perfect record, but there is correlation where during the weak Aleutian low, we have our faster growth rates. So consistent with the weak Aleutian low, there's no clouds, there's no wind, surface water mixing, there's less plankton, it's sunnier, more light reaches the seafloor, and the algae can grow more. So this is kind of consistent with our interpretation. We also looked at cloud cover, which is, again, related to the Aleutian low. And even the Aleutian low and cloud cover is not a perfect agreement, but between the two of them, we can pretty well explain the growth rate measurements that we were able to see in the algae. What was really neat about this record, though, is then we compared it to salmon fish catch. And it's a complicated graph, so I'm going to walk through it with you. And at the top here is proxy salmon abundance measured in delta 15N lake proxy record, because the nitrogen byproduct of salmon is very, very different from the nitrogen that's there otherwise. And then we have the official catch record kind of towards the end of this plot. And then we have the PDO plotted on here. And then for comparison, we have our algal record down at the bottom, and we have the number of samples that have gone into this reconstruction. And obviously, because not all our samples are 300 years old, we have more samples towards the present than we do into the past. And what we find is that the delta 15N proxy record of salmon abundance actually tends to match our Aleutian low record more than it matches the PDO. And more recently, people have really attributed the PDO, changes in seawater temperature, as a driver of salmon abundance. And what we're showing here is that's been true recently, but historically that's not been true. Historically, the Aleutian low is a better predictor of salmon abundance. And that makes sense if you think about it, because the Aleutian low is related again to the mixing in the food. So the salmon actually probably like it when there's more productivity in the surface waters and there's more food. So this was the first time that we had shown that the salmon record is not always related to the PDO, and in fact, it's also probably related to their food variability driven by the mixing of the water. So I'm going to switch to part 3 of the talk now, and I'm showing you a pretty white figure, and the point here is there's not a lot of data, and what I'm going to try and do is fill this in. So these are your gridded data. They are the NOAA's Extended Reconstruction SST, and then your Hadley I SST data sets, plotted for a grid that encompasses a nice little box around the Aleutian archipelago. Not the whole kind of area, but around where my islands were. And this is the number of the ICOEDS observations that have gone into these data. And you can see that going back to about, as I said, about 1950, 1960, there's quite a number of observations. But when you go back prior to that, there's not a lot of data. So we still have gridded data products that are determined by interpolation from extent locations. So these data are not generated from these locations. They're generated from modern relationships, statistical relationships of seawater temperature with other locations where we do have measurements. And you can see that between the two different gridded data sets, sometimes they agree really well, and sometimes there's quite a lot of disagreement between them. So now is an example of where we can use the algae to try and fill in this data gap and extend the seawater temperature record further back into time. We can do this with the algae because the magnesium-calcium ratio in the skeleton serves as a paleo-temperature proxy. When temperatures are warmer, then you're going to have more magnesium incorporated into the skeleton, and when temperatures are lower, you're going to have less magnesium incorporated into the skeleton. This is a ratio of algal magnesium-calcium over time versus seawater temperature on the right, and you can see that they both very clearly show your seasonal cycle in reflecting the changes in seawater temperature. It's not a perfect fit. There's a lot of other things that are also involved here, but we can see that there's a very strong relationship between temperature and algae magnesium calcium. So what we did is what our goal was initially was to quantitatively reconstruct seawater temperature. And kind of to date, people had been generating a time series record and then just interpreting the periodicities in it. But we wanted to know absolutely how did temperature change, and to do that properly, we wanted to have our uncertainty on it. So we performed a replicated calibration experiment from the collection that we had used for the growth study. We took specimens from 3 islands, Akun Island, Mchitka Island, and Attu Island. From each of the 3 islands, we had 2 specimens, and then each of the specimens, we had replicate transects. And so again, from these specimens that we had already measured the growth, we used laser ablation, inductively coupled plasma mass spectrometry to measure the magnesium-calcium ratio over time. So these are our replicate transects from one specimen, our magnesium-calcium values, and we lined it up so that the minimum magnesium-calcium value was our winter month, our coldest winter month. Our maximum magnesium-calcium value was the warmest summer month, and then we used that for each winter and summer to develop the growth chronology over the entire duration of the record. And with this one, we were actually able to extend it a little bit further back into time to 665 relative to our growth reconstruction. One of the first things we found, though, and initially this is just using post-1960 data, is that there was island-specific variability. The winter temperatures were pretty similar between the different, the winter magnesium-calcium values were very similar between the different islands. But when we looked at the summer magnesium-calcium variables, there was a bit of a difference between the different islands. We're not sure why that is. We haven't been able to figure out what's driving that kind of island-specific variability. But what it means is you cannot use one calibration equation developed on one site and apply it somewhere else. You need to be able to develop island-specific curves. And so here we use the gridded OISST data. This only goes back to 1981, but it's in higher resolution than some of the other gridded data products. And we developed a transfer function where we can take the amount of magnesium calcium in the skeleton and then convert it into an absolute temperature. And we did this for each of the 3 islands, and as you can see, they ended up with 3 different transfer functions. And that's really important because sometimes people will take their calibration equation from one location and then apply it somewhere else in a different location to get temperature. And we're showing that you can't necessarily do that. The error is going to be a lot bigger. And then to get that error, we used our replication, and we reduced the standard error of regression with only 1 specimen. It would have been plus or minus 0.9 degrees Celsius. But by using multiple specimens, we were able to get it down to 0.45 degrees Celsius. Now, plus or minus 0.45 degrees Celsius is really still not that great. And there's a lot of other variables included in here. One of the things is we're calibrating the algae, which are, again, maybe 10, 15 meters deep, to the gridded SST, which is the surface skin of the water. So that alone probably explains a lot of the variability. The water at 10, 15 meters deep is actually within the range of the thermocline variability. And so the water that's being measured at the surface for the gridded SST data is not the same temperature water at depth. But there is no in situ temperature water in the Aleutians, so we were working with what was available. And we're trying now to constrain how much of that variability drives the uncertainty to see if we can work to reduce that uncertainty. So what we did is with those 6 specimens, we then reconstructed 45 years of data to see how well we did. We verified it against an air temperature record from a nearby island. It's not even the same island that our algae are from, but it's close by. It was I think it was an army base, and so they have been measuring air temperature with a thermometer. And then we used the HADI-SST, which for the past 20 years is very similar to the OI-SST, but it extends a little bit further back in time. And again, 1960s when they really started to pick up the number of observations in the area. So we trust the data a bit more post-1960 than we do pre-1960. The gray is the error of the reconstruction. Again, I wanted a quantified reconstruction with error. And you can see that they agree pretty well, capturing the decadal, multi-decadal trend. Another way to verify is to do a spatial correlation. And this is the algal data set from the Aleutians with the gridded data. And you can see that we significantly correlate at the site of our algae with the gridded seawater temperature data. And what it picks up here is the Pacific Decadal Oscillation horseshoe. And if you take the gridded data set and you compare that, you'll find that the similar pattern shows up. And that's because with the PDO, this water here will be warm, and then when it flips, this water here will be cold. So this is what we expect to see in the Aleutians, and what happens is this water here gets transferred along the Aleutians via the fast-moving Alaskan stream. So we have confidence that kind of on the maybe interannual decadal scales with a decent, but hopefully we're starting to get it lower uncertainty, we are capturing changes in seawater temperature in the Aleutian archipelago. So the next step then is to go to our multiple specimens going back into time and doing our multi-centennial reconstruction. So again, to do this, we only focused here on 2 of the islands. We looked at Attu Island and we looked at Mchitka Island because those are where we had the longest records. The records from Akun Island were much shorter, so we didn't see as much value in trying to do the reconstructions. We analyzed the magnesium calcium for all the specimens, and we replicated it wherever it was possible. We averaged it for the specimens, and then we averaged it together for each of the island. Because the error of regression on our calibration depends on the number of specimens, we kept track of the number of specimens, and that way our error is actually a sliding error. So the more specimens we have, the more confident we are in that uncertainty. The fewer specimens we have, the less confident we are. And so we are able to build that into our reconstruction. And so here I've plotted the 2 different islands over time, going from 1665 to 2003, the number of specimens for each of the island reconstruction, and it's been filtered with a 36-month filter, low-pass filter, with the uncertainty based on the number of specimens for that island. And then I've also shown the first plot that I started off this part with the HADI-SST and the ER-SST and the number of ICOADS observations for the grid cell that encompasses these islands. So right off the bat, we have actually pretty good agreement between these two different islands. They're showing the same multi-centennial trend. We have warming starting from about 1665 coming up. There's a peak in around 1800. It cools again, and then it comes back up. We only have data from Amchitka Island for this point in time, so we're hoping at some point to get more specimens to fill in and see if Attu or other locations are recording this warming as well. And then again, down here, we only have one specimen, and here we have two specimens, and then here we have one from each island, but they overlap within the air. This could be some kind of a specimen-specific, something happened here, so we'd like to see this reproduced. But you can see this general trend, and then it matches the ERSST and the HUD-ISST very well in the recent. There's a couple spots where it deviates, but again, in these locations, there aren't really that many observations. So who's to say that the gridded SST are actually doing a better job than the algae are? And it also could be changes in the thermocline structure or something else that's changing here that's kind of reducing the agreement between the different records. And the kind of the big point from this is what we're showing is this 1.2 degree increase in seawater temperature since 1665. So next, of course, we want to try and determine what's causing the increase in temperature. This isn't necessarily consistent with some of the other oceanic or atmospheric records. So we looked at two different variables. We looked at volcanic activity using the volcanic explosivity index at the top. And so we've plotted volcanoes here with an index that are greater than 4. And these are only for kind of the top half of the northern hemisphere, volcanoes that would impact the Aleutian archipelago. So they're plotted across here. And then we've plotted some of the solar activity, the Maunder minimum, and the Dalton minimum. And what we find is neither of these do not relate to driving the seawater temperature. With the volcanic activity, it might be that a year or two after a volcanic event, there is a short-term cooling, but it's within the uncertainty of our reconstruction. So we're not able to tease that apart at this point. And if solar activity was driving the record, then we wouldn't have this disagreement between the cooler temperatures here and the warmer temperatures here.
Yep. Volcanic activity, did you give more weight to volcanoes that are like right on these islands? For instance, like the Cleveland, Mount Cleveland one?
We didn't give them more weight, but I looked at them and we don't see, there's nothing shows up.
Do you see any Teflon layers in the algae?
We don't. That would be fantastic. But we don't, no. No, the algae is perfectly clear. We didn't use specimens that had stuff in it. So we did a lot of quality control. There's a lot of big algae that are still sitting in the lab that never got analyzed because they've been eroded or bored or were otherwise beat up. So, and it could be that there is the temperature there, but again, if temperature only decreased half a degree, it's within the air, so we just can't see it. And that's something that we get more specimens we might be able to address going forward. So what we're working on now is trying to understand if there is any periodicities. The PDO, the most recent PDO, this big increase right here matches the 97 PDO shift event. There's a decrease in here actually that could match the one that's kind of hypothesized to have happened in 2000, but we don't capture the 1945 or earlier events, and there's very little relationship between our record and other PDO reconstructions. So although we are at the edge of the PDO, we're not really seeing any type of the PDO in this record. And so just for the last part, I'm just going to kind of show you this one slide, but the other thing about these algae, and I've kind of alluded to it, is that they have this amazing kind of high latitude distribution. I spent today talking about this species, Lathromorphum nereostratum, which is found along the Aleutian archipelago, but its sister species, the Lathromorphum, which we've been also doing a lot of work on, is found all the way from Maine, up the coast of the Northwest Atlantic, through the Canadian archipelago, around Greenland, and then all the way through the Arctic, the edge of the Arctic Circle. So we're kind of just really scratching the surface of the type of records that we might be able to get out of this algae. I was up in northern Canada and Nunavut this summer collecting, and this algae can actually grow under seasonal sea ice. And if you think about what's going to drive growth rates when you have sea ice, it's going to be the sea ice extent, the duration of the sea ice and perhaps the snow on top of the sea ice. And one of my colleagues has demonstrated that he can capture sea ice variability in these algae. So now we have a chance of developing an annual scale proxy of sea ice, which is a really important contribution to understanding how these high latitude environments are changing. So kind of just a summary and to wrap up, just to go through kind of what I talked about. Clathromorphum, it's a long-lived coralline alga. It documents changes in the environment in its skeleton. And we also know that it has a distribution that can fill the data gaps. It's long-lived, that longer specimens are out there. We just need to have more extensive field campaigns and people who want to go diving in crazy cold water. And then also it has a skeleton that so far has appeared to be fairly resistant to diagenesis outside of those conceptacles. The Aleutian low is the regional factor that drives light levels reaching the sea floor in the Aleutian archipelago, and that is what strongly drives the algal growth rates. And that the algal growth rates track salmon abundance, and thus we're showing that the salmon abundance actually relates more to the Aleutian low going into the past than it does to changes in temperature variation. And then we also know that if we do replicated calibrations, we can permit absolute temperature reconstructions with a quantified error. And that doing such a reconstruction going back into 1665, we know temperature has increased by about 1.2 degrees Celsius. And that it's not forced by volcanic activity or solar variability. So it's probably related to the climate patterns that are kind of unique. The intersection of those climate patterns unique to that area. And that's what we're working on now to try and tease apart. And then the abundance of Clathromorphum at the high latitudes yields really the potential for unparalleled records in helping to fill in these specific data gaps that we're missing in kind of understanding the high latitude change. So I'm going to leave it there. Thank you for listening, and I'd be happy to take any questions.